Next up, we have Florian. Florian, good to see you.
I cannot share yet because I think somebody else is sharing. Can you hear me?
We can hear you.
Now I can share. Wonderful. Let me start by telling you how much I regret not to be there in person. I was already since Aviv's talk hanging on Zoom, but I'm teaching here in Cold Spring Harbor. And then yesterday I was sitting for quite some hours in an airplane that never started. So you can now hear my talk, but fully selfishly, I much regret not being around all of you after my talk. Okay. If the AV people could make me see the audience, that would help me feel less than me talking to my own mirror image in front of me. Amazing. Perfect. Thank you very much. Okay, I am gonna tell you about a bunch of things, but I would like to start by reflecting on how I see AI being used in the life sciences, and that is mostly, or how it is there to support us all. And this is mostly making a statement about the microscopy sub-community, I would say. There was maybe 18 months ago a special issue in Nature Methods that asked what will the next 10 years bring? And many people had many opinions, and we were one of them. And we pointed out a relatively obvious thing that methods research needs, of course, the data from the life sciences so that AI can come up with methods that will facilitate the rate of scientific discovery. But there's one very important thing, and that is that there's tool development really needed. And in Janelia, I mean, this is one of the few places where this is taken very seriously, I would say. A method is only as good for a biologist or life scientist as the enclosing tool makes it usable. And so in AI for Life, which is a European-funded project that I was running together with Anna Kreszuk for the last 3 years, we do lots of these things. I don't want to go too much into details, but the idea really is to support the integration of deep learning into a growing number of open source software tools via the BioImage.io model zoo. So here we can take methods and put them in the standardized form into a zoo of models that then can be used in these open source software tools and in this way be brought to life scientists. But I would like to focus on one other aspect of this relatively large project, and that is open cores. So for 3 years we had open cores. This is a bit of an outdated slide for only the first 2 iterations, where a total of like 160 or so people by today applied to get support to solve a problem where they think deep learning should make things easier. And I will not go into all the details. You know, we select the projects where we also think this might be likely, and at the end, kind of write up what we did together with these life scientists. But the big lesson learned is many people want help, and at the end, almost everybody needs rather basic image analysis support, and that's sad. And I think one of the main reasons is that we really have to work on this method to tool transition to be able, even with us helping, to kind of put the latest methods into the hands of life scientists and really make their analysis problems easier. Next to this European project, also in our institute, I work at the moment in Milan at the Human Technopole. We have, it's a relatively small place at the moment, just shy of maybe 500 people, and we will grow to maybe 1,000. We fall into 5 different centers, you can think departments, that I show here. And the really important design choice is that the scientific activities, they are all kind of make heavy use of centralized facilities. And in these facilities, I was tasked some years ago to set up the image analysis facility. And so also here, we really kind of put this gradient model into play, where we do methods research, method to tool transition, and then have this facility that kind of serves, in our case, all people on campus. But on top of it, we also have these facilities nationalized. So every publicly funded Italian researcher can write a relatively easy application and then come to us and use our facilities. And the advanced imaging center at Janelia was really inspiring us of how to set this up. So I will speak a lot about methods research in the remainder of my talk. Then just a few words about this research software engineering method to tool transition. This is much bigger than our institute. There is image.sc, I hope some or many, all of you know it, where a huge amount of open source tools are sharing a joint forum. So if a user that has image data in front of him or her would like to analyze it and asks a question at image.sc, all these different tools have representatives there that can help you find good ways how to analyze your data. Then we also organize from images to knowledge. So there was like more than 100 nationalities of research software engineers that came together to discuss how to best do this method to tool transition. We teach in Woods Hole, we teach in Cold Spring Harbor. The reason why I am actually not with you is because I had to teach until yesterday and then the flight didn't go. And we also go to computer vision conferences and organize workshops there to kind of really bring the nerdy CV, ML, AI community and the life science community closer to each other. And then I just flashed this very quickly at Human Technopole. Also, only the image analysis facility is rather sizable. So we will grow to this number of people and all the check marks are hired or in the process of being hired. So again, speaking about this in Janelia is funny because in any other place, in almost every other place, this is an enormous quantity, but Janelia has us covered, I guess. Okay. Let me move on to the more researchy part of this conversation, of this talk today. And I will do a bit of archaeology. I will go back to the 2010s and speak about some methods we did back then that are still used, but they are old. And so I will point out that these are point predictors, and then from there we will go into more modern things. So I will keep this really, really short, also in the interest of time. The idea of content-aware image restoration was the following. You go to a microscope and you image gentle, not to destroy your sample, and you get to a certain signal to noise. But if you were to image with more exposure, then your sample dies quicker, bleaches quicker, has all kinds of problems, but the image quality is higher. And we thought, if we do this paired, then we can now train a neural network. And it was a simple unit. Back then, that was state of the art. And then get worse, so lower signal to noise ratio data in, and kind of can computationally see what the higher signal to noise ratio image was looking like. And to cut the long story short, you see here on the left the sea of noise as it comes in 3D from the microscope, and then on the right the cleaned up image. And I think it's obvious that if you want to count these objects, segment these objects, it's all easier. So downstream processing is made more simple. And while this is relatively easily done, a bit of work on the microscope and a bit of training, very soon we realized that people are maybe for good reasons lazy, and sometimes high quality training data cannot be imaged at the microscope. What do you do then? And the idea that we had back then was, let's take single noisy pixels and put them on the ground truth side of things, and now train a network to kind of in-paint this pixel and supervise by the noisy pixel here. And, I speak about it many years and I can really not hear myself talk about it, but the basic intuition is that we use an inductive bias. We heard about inductive biases this conference already. An inductive bias of noise. If noise is pixel-wise independent given the signal, so per pixel, the noise is kind of an independent coin flip, then the best the network can do is to predict the signal itself. And we do that parallelized so that it's a bit faster, and lots of engineering goes into it. And I cut, again, a long story short and just show you if the images that come from the microscope are rather noise-ridden, then after noise to void self-supervised training, no ground truth needed, just a body of data to train on and to predict on as well. All this Poisson noise and Gaussian noise gets removed. Okay. that was just a reminder, and some of you might never have heard about it because you don't even work with microscopy images. So that are methods that are still used quite heavily because they are also very convenient, don't need much to be run, are very fast. But the reason why they work is that with neural networks, we have a tool where we can learn strong structural priors from data. So the network learns how the things you want to denoise actually look like. And they can now do this inverse process of denoising, aware of how the things in the images look like. And that is really giving the competitive advantage here. And then, yeah, and these structural priors help to find sensible solutions for inverse problems. So I said both already. Now we come to the point predictor part, and this is an important point. One thing that I dislike about our methods from the 2010s is that they give you the impression that they know how the denoised image looks like. And that is just not true. Because every noisy image can be the result of noising a whole number of denoised images. And there is some uncertainty. And this uncertainty is just like put under the rug. And let me bring a very simple example that I hope helps for your understanding. If I roll a die, I can either roll a 1, 2, 3, 4, 5, or 6. And if I ask you to minimize, so you have to guess what I will roll next, but you should minimize how much you are off. This is essentially what we ask the networks. Predict the non-noisy pixel. Then the best you can do is you can say 3.5. So you will always be kind of in a quadratic loss. You will always be closest to what will really happen next. So you minimize the quadratic loss by guessing 3.5. But you will also notice that 3.5 is very unlikely to really happen. I never rolled a 3.5. And so the same thing happens with the denoising networks I showed you before. If there's uncertainty, if you get an input that is, I mean, my drawing scales, I think, yeah, can be improved. If there is uncertainty in the input, the network will give you something that is actually the average of all possible interpretations of the noisy input. And it will give you this average. And so if you ever looked at noise to void, care, or a whole bunch of other methods by now that work with point predictors, with networks that give you a single answer and are trained with like classical losses like L1 or L2 losses, what you see is that in places where there is uncertainty in the input, the prediction looks washed out and blurry. And that is because you essentially have the sum of different possibilities being given to you in one go. And we don't like this. And so, yeah, we have denoising methods that don't suffer from this, but I will jump over all of them and introduce a different task that we solved recently, but also solved it with a different type of model or method that doesn't predict a single averaged answer. And these models are called posterior models because there we learn a whole posterior of possible interpretations from which we can later sample instances and can say, oh, it could look like this, or it could look like this. So let me jump right into this. This is the main part of this talk. I hope, I see you all very small. It's clearly too small to read your faces, which I usually heavily use as a cue for how ununderstandable I am by now. If some of you feel really unhappy with my talk, just stand up and look angrily in my general direction, then I can maybe change my tune. If you go to a microscope and you do, like not fancy microscopy, you just want to get a good fluorescent microscope image from your sample. You label different structures with fluorescent dyes or fluorescent proteins, and then you do multiplex imaging. You excite one fluorescent protein species or a dye, and you collect all the light from the emission, and you kind of filter out something that blocks out most of the light from the other channels. And you do that temporarily multiplexed, and then you get 3-channel image, maybe have a bit of bleed through, everything is okay. Now, if you wanted to do that not with 3 colors, but with 5 colors, everything becomes a bit more complicated. The microscope might not have enough suitable filters in the filter tour. It just gets a pain. But since we can now train really strong structural priors, we thought, maybe we can actually get to convincing solutions, even if we put 2 structures that look sufficiently different into one channel, acquire it, so to say, together in one single image, and then use a suitably trained AI method to tell these 2 channels apart. And then we have a free channel, which we can either not use and image gentler, less photon exposure, make better use of our photon budget, or we can image a thing more. And so I will show you, just so that you see that this is not completely ludicrous, I will show you an input that actually contains 2 different structures. And you can look at it and maybe quickly in the next 2 seconds form an opinion of what these 2 structures might be. And then I show you the result of Ashesh's method, which we called microsplit at the end. So it is not only working if things lie neatly next to each other, that it carves one thing into one partition and one in the other. These things are really heavily overlapping. And it's really, I found it very surprising to see how well it works. I mean, you don't see that yet, but I will show you later. So also at this resolution, you know, it looks really exactly the same, and we will drill down here a bit more in the next few slides. But yeah, so this column is the temporal multiplexed ground truth, and you can see that you don't see a difference, at least not on this slide. So what we need here is a strong structural prior, and so this is maybe my most technical slide. This is not units anymore, obviously. So what we use is something, we use variational architectures, something like VAEs, if some of you know. But we also modified them a bit. So first of all, we use so-called ladder VAEs. They have a hierarchy of different levels. And at each level of this hierarchy, we do one more crucial thing. We start with an image patch that we would feed in the past to a unit. Now we feed it to our hierarchical VAE setup. And then when we start processing it in the next layer, we kind of downscale this image like we also do in a unit. But then we thought, once we downscale this image, this patch that we fed is now smaller. This patch came from a microscopy image that is way larger than the patches we train on. So we could now, in principle, pull in more lateral context of where this patch comes from in the big surrounding data set. But we also pull it in, of course, at lower pixel resolution, so also in a downscaled way. And so we keep the patch size, if we go up the hierarchy, constant. And we feed more and more lateral context at lower and lower pixel resolution. And that is nice. So we give more context. That can only help. But there's another thing that happens. If you have K is the width of your patch. So the patch is K square in size, let's say. No, 2 to the K. 2 to the K in square in size. Then after K steps, you are left with a single pixel, and then your hierarchy necessarily has to end. But now that we pull in additional lateral context, after K many hierarchy levels, we can still go on and now make the image indeed smaller for another K hierarchy levels. And so we can make a way deeper, larger network that has a bigger capacity and the potential to extract really a maximally informative structural prior of how the things look like. In this case, that we don't want to denoise, but that we want to kind of split into different image channels. So we essentially kind of put 2 of these hierarchies on top of each other. Yeah, that was the technical insight that I think really enabled this method to perform very well. And now I will bring a few more footnotes, side notes, details. So this is another image where I chose one that is relatively easy also for humans to kind of split apart. You see like liney stuff and dotty stuff, so puncta and lines. and we would like to kind of tell them apart. But what you can also see is that the image itself is rather noisy. And since denoising is our everyday business for many years now, here a cartoon of what we really want to do. We want to give the network a superposition of two structures that we want to pull apart that is very noisy. So we can do an easy job at the microscope, image gentle, etc., etc. And then we want to supervise with multiplexed images that are also very noisy, right? So also the training data should be allowed to be very, very noisy. And then there is some footnotes about, read the paper. We need noise models, but everything benign, easy to generate from your data. And then we want to predict a denoised split channels. And so let's see how that works. So this is the training data. In this particular example, we take this training data that is very noisy and we just like do a pixel-wise summing. So we get this like merged images that we now use for training together with the training data. And once we predict, you see that the self-supervised denoising did its magic, and you now get denoised versions of the original inputs, which, okay, these are not inputs. You get denoised version of the split of the superimposed structures. And if we compare that to non-gentle high SNR images we acquired just to give you the pleasure of comparing these images, you see that you don't see a difference. And that makes us happy. So I zoom in a bit more so that you see also here is a histogram. The red one is the prediction and the other color is the ground truth. So everything seems to work very well. But there's one more really important message. So even if you are dozing off, please wake up again. This is really the key point that I would like to drive home. Now, so far, using non-point predictors, using posterior models, didn't buy you anything extra. But now we have an input, a noisy superposition. We can now sample from the posterior. So we can say, give me different interpretations of how these two structures, if you kind of would have imaged them in two different channels, would look like. And since it is not, the input is not very ambiguous, the two samples I show here, they look rather similar. So I subtracted one from another just to show you that there are actually differences here. You can see that some puncta are brighter or darker. Sometimes one might not exist. But what we often do then is we sample 50 of those posterior samples and we average them, which sounds now a bit counterintuitive. The reason for this is that point predictors are very smooth and often very easy to analyze. It's just in places where the ambiguity is very high, they over smooth and there is no way back. Here we can always go back to the individual samples. And we do that for one particular reason most. And that reason is that everybody that uses this would like to know, nice to have this prediction, but can I trust it? Does this actually make sense? Is this actually what my sample looked like? How off am I if I had high SNR ground truth? That's the question. And we went through all the pain of generating this high SNR ground truth so that we can plot it here on the y-axis. But in any normal circumstances, you don't have it or the whole method makes no sense, right? So this question is really in everybody's mind. And now we proposed the following way out. We asked, how different, what's the variability of a pixel over these 50 samples we averaged to this MMSE result? And we can plot this on the x-axis. And what you see here is that the inter-sample variability scales linear with the true error, which you usually cannot assess, right? But if you do that on a small subset of your data with relatively little work on the microscope, then you can go and you can put this in your paper and you can convince your reviewers that applying it to more data of the same kind allows you to estimate the error you really made and maybe discard places where this error is too high. So we call these models that have this property that they are calibrated. You can show that your model is calibrated and make your reviewers happy. And yourself too. I mean, that's maybe even more important, right? So this is now a bioRxiv preprint. I'm almost done. This will go fast. We don't only split 2 channels apart, but also 3 or 4. So if you look at this, there are 4 structures fluorescently labeled and on top of each other. The training data is very noisy, but you see that the predictions are not, and that is very nice. And we also give the possibility to compute these heat maps, where whenever you see something lighting up here, then you know that there is maybe too much uncertainty in the input. You should not analyze this part of your data. So if you look at this place here, God knows how many puncta there are, and also the samples from the posterior, they are quite diverse. There's 1 enormous puncta or 2 or 3. So here you see 10 samples from the posterior of only this region of interest. And yeah, the variability is really high. It points it out here. You could discard this area if you wanted to count whatever these puncta are, expression events or something like that. We did that on a total of 29 different tasks for 2 channels, 3 channel and 4 channel, unmixing in 2D and in 3D. Everything works in 3D by now, which is really great, natively, and the results are amazing. So PSNR, everything above 30 is very high quality. So yeah, I was expecting it to work good, but it works better than I ever thought. And the take-home message of this part of my talk is the same as before. Structural priors help you to do inverse problems really, really well. But then if you use variational networks, it is really nice for error estimation and for estimating the uncertainty, the data uncertainty in your inputs, which I think...
Florian, I hate to cut you off, but you're a little bit over, and I want to make sure there's some time for questions. So could you wrap up in like the next 30 seconds?
Luckily, this is my last slide. Thanks a lot. I started early maybe. Okay, whatever. I'm sorry for that. So posterior models are good for at least this case. And then what comes next is something I will tell you the next time I'm in Janelia. And thank you very much.
Awesome. Thank you so much. Time for a few questions.
What comes next? Hi, Florian. What comes next?
I might have a slide here. So I think AI will find curiosities in your data. So this is the Waddington landscape. This is what you can observe. But what you are really interested is, what my director always asks is, how does it work? And the how is best explained by understanding the underlying principles. So this is also a drawing from Waddington. And this is not only true for what Waddington had in mind, but across scales. And I think we will have to use AI in some way to point us at statistically interesting points in multimodal and data that spans different length scales. And yeah, my plan was to tease you with this and then have a whole day to discuss it with all of you. But unfortunately, I'm stuck at Cold Spring Harbor.
Hi, Florian. I have the obvious question. How many images do I need, and what should they look like for this splitting, for the channel splitting? So what should my data look like?
You just do your regular multiplexed imaging experiments, and you make me more happy, but maybe yourself less happy if the data you acquire is rather noisy, because then the method excels even more. And depending on how complex your task is, you might need more or less data. But usually, if you do 1 or 2 imaging sessions and you come back with maybe I don't know, between 1 and 3 gigabytes of data, that should totally be enough. And then the next nice thing is that if you don't have enough data, then you will see it. You will see that the loss is not yet flattened out. And it will naturally tell you, maybe go to the microscope another time and give me 5 more volumes or so. It's all not crazy. You don't have to spend 3 months at the microscope to get petabytes of data. Not at all.
Great, thank you.
Last question.
From your experience with like the community and the microscopic analysis, what is the most important modification between off-the-shelf tools that people develop for images in like computer science and general tasks like on ImageNet, for example, that you would need to adapt to handle well with our biological data?
To make it work in the first place is 3D plus maybe time and channels. That is a big difference. But so that is the reason why you cannot even run it oftentimes, because it expects a 2D RGB input, right? And it's not what we have to offer. But in reality, I think it's also a bit of a different mindset. On natural images.
No, of course, I meant the deeper layer, not the technical tools. If you have some deeper insight, or maybe there is place to mind chip to different architectures completely, and we shouldn't rely on these models.
Yeah, we should at least adapt it to the input shapes that we want to give, right? To the dimensionality we want to give. But I think we also have to adapt our mindset and think more careful about scientific data, because you don't want to screw up, right? This is also why the calibration I showed today, I think, is a really important step, that on natural images, who cares?
No. Okay. Thanks so much, Florian.